Hello, hello. Hi, everyone. Thank you for joining this live session. We got 8 minutes more before we start our webinar session. Today will be me, Shikin, presenting a topic on microorganism under our Biofloc system. This one is specifically on the seawater and the Banami species. Everyone can hear my voice. Is it too loud or too slow? We still waiting for more viewer for this live session. I don't know the voice clear or not. Hai Arasan Chandru. Hey, hello, good evening, Hearty Habit. Where are you from? We will start our presentation in less than 1 minute. Okay, it's already 8:30 here in Malaysia. So I will start the presentation. Thank you for taking some time to join this webinar. First, I will introduce myself. My name is Shikin. You might already know me well from our YouTube and Facebook video. My role with this company is as a marine biologist, where I am responsible for the operation of Biofloc shrimp farming here, from the nursery stage, feeding, solid waste management and grow out observation for the shrimp. I am also involved with a part of micro operation and consultation service. We have met clients with a varied background that want to pursue this aquaculture technology, whether RAS or Biofloc. What we do here is helping those people to manage their farm based on our own experience. We have also tried few design before we come up with this current Biofloc and mud-crab farming setup. So we somehow know what are the good and bad of the system. We have done many webinars before. So if you haven't watched the previous one, you can scroll into our YouTube channel. Today webinar is about microorganism under the Biofloc system. Just a general idea on Biofloc. Some of you here might be very familiar with the term Biofloc. We have done much content on Biofloc on our YouTube before. You should check on our YouTube if you have not got a clear idea yet on Biofloc. So what is Biofloc? Biofloc is the aggregate or flocculation of good bacteria and microorganisms in the culture water. These microorganisms are heterotrophic bacteria, algae, fungi, ciliate, flagellate, rotifer, nematode, metazoan, and detritus. But not everyone can have all of these microorganisms inside their biofloc system. The presence of this microorganism will help to maintain the water quality, maintain biosecurity, support high density shrimp and help to reduce water exchange. In the aquaculture industry, molasses is a product that has been used widely. The use of molasses or any carbohydrate as the source of carbon in biofloc will help to encourage the development of heterotrophic bacteria. Sometimes, when the farmer has high ammonia issue in the system, they will dose the molasses so that the bacteria can duplicate and work efficiently to remove the ammonia compound. Heterotrophic bacteria mechanism to remove the ammonia can be seen from this diagram here, over here. Ammonia in the water will be assimilated into the bacteria cell. This cell later will be a form of protein source for the shrimp when they consume the floc. Moving on to microorganism. Microorganisms, especially bacteria, are usually rich in protein, which can make up to 65% of the total dry weight. They also have more nucleotides compared to traditional fish meal. The application of this nucleotide supplement in aquaculture diet is relatively new compared to mammalian and human counterparts. These nucleotides have been added to infant formula for many years, but the knowledge on the application of nucleotide in aquaculture is very limited. Nucleotide content in the bacteria can help to improve survival and growth under stress. Also, there is a study that reported that Litapinus vanae fed with 0.04% diet nucleotide could elevate non-specific immune response, resistance, and growth of this species. Surprisingly, we can get all this benefit through the microorganism composition in Biofloc. But be careful, too many bacteria inside the system would not preferable as it will cause another potential problem. Beneficial bacteria, they improve the aquaculture yield and aquatic product quality by suppressing microorganism activity. There are three major approaches for beneficial bacteria to suppress harmful aquatic microorganisms: competing for nutrient and space, producing inhibitory product for direct antibiotic effect, or disruption of signaling, such as quorum sensing and stimulating the host immunity. Quorum sensing is a process of bacteria cell-to-cell chemical communication that respond to signaling molecule called as the autoinducer. The autoinducer is a compound secretion by the bacteria cell. This biostatic effect suppress harmful microorganism-induced aquatic animal disease and product spoilage to advance the yield and quality of aquatic product. This is roughly how we can prepare or start making the Biofloc system. Prepare the water, CN20 dosage, and floc monitoring. The details on how to make this happen have already been explained on the previous webinar on the nursery review. These are the materials you need to promote the growth of heterotrophic bacteria: pellet, molasses or the carbohydrate, and also probiotic. Make sure you are dosing CN20, carbon to nitrogen ratio 20, for fast bacteria growth. From the clear tap water or filtered seawater, you can see the water will turn into brown color. After 1 to 2 days of dosing CN20, you will have young or unmatured floc, as you can see in this picture here under the microscope. Young floc will look dispersed with fewer bacteria and dominant with heterotrophic bacteria only. Only after 1 to 2 weeks, the floc will become mature. When they are mature, you can see more compact floc under the microscope and has more microorganisms. At this time, your system will also contain autotrophic bacteria, which are responsible for the nitrification process. You only need 3 things to observe the microorganism. First, compound microscope, slide and the cover slip, and also plastic dropper or pipette. To prepare the sample, you need to take the sample in a beaker and wait for a while for the floc to settle down. You can also choose to take the floc that settles down after your Imhoff cone measurement. After that, you can use a dropper to take out the floc from the bottom of the beaker. Next, you need to place only a bit of the sample on the glass slide, and then you can observe under the compound microscope. The next step is we need to count and record what microorganism and how many of them present on the slide. This process usually takes about 10 to 15 minutes, depend on how good is your microscope skill. This is the example of our microorganism count sheet that we use here. Here on our farm, we do the microscope count twice a week. It is also an important parameter to look at in Biofloc. The change in the microorganism count is also a type of signal that your biofloc is changing their characteristic or changing to young or old floc. Sometimes, even if your floc is already considered as old floc, they can change back to young floc if you keep removing the solid from the system. These are some footage of live organism under a microscope. The biofloc system is in a good stage when you have a variety of organisms on one drop of water. Take note that these microorganisms, they will have many shapes and species although they are categorized under the same type. So here is the video of variety of organism under one slide. This one is the ciliate, stalk ciliates, and rotifer. Okay, so now we will go through one by one of the common microorganism in seawater biofloc. This is the free swimming and crawling ciliates. The ciliate is most dominant and you can see it every time you observe the floc under a microscope. They usually prey on bacteria, so it helps to produce low total suspended solid or TSS. We know that high TSS will increase the turbidity of the water, which will disturb the visibility of the shrimp itself. The presence of this organism indicates a good quality sludge and is typically found in young to medium aged sludge. How you can identify whether your sludge is in what age? It's by observing the sludge color. Darker color indicates older sludge age. The availability of the ciliate also helps to facilitate the population growth of the rotifer. If there is no ciliate in the system, it is quite hard to get rotifer inside. However, the limiting factor of ciliate development is when there is a shock lot of toxic materials, lack of aeration, and excess sludge. Next is stalk ciliate. Stalk ciliate is often found growing on stem or myomas and can quickly retract like a spring. Stalk ciliate protozoa help to reduce the turbidity of biofloc water, and the moving action of their anterior cirri and myomas indicates the efficiency of stalk ciliate to remove excess floc. The springing action of the myomas produce a water vortex that draw bacteria into the mouth opening. However, not all stalk ciliate protozoa have a contractile filament. Stalk ciliate and rotifer both need a stable biofloc so that they can survive. If there is no observation of these two organisms in the biofloc, meaning that the biofloc system is not stable yet, it might take a bit of time before it becomes stable and ready for the shrimp. Rotifer or wheel animalcule can be in the form of various shapes and over 2,200 species are identified. Rotifer are frequently associated with biofloc fulfilling some fundamental function such as floc fragmentation, consuming bacteria, and produce mucilage that helps to the new floc formation. This organism can contain between 54 and 60% of protein. Their feeding method is by filter or suspension feeding. The rotifer, they crop bacteria and release sticky secretion that coat fine solid, making the solid more compatible for adsorption to the surface of the floc particle. They also recycle nitrogen and phosphorus by cropping and digesting bacteria and protozoa, and also excreting nitrogen-containing and phosphorus-containing waste to the bulk solution. Rotifer is important in the biofloc system as it provides amino acid and saturated fatty acid, which help to improve shrimp survival and growth. The generation time for rotifer can take several weeks. However, the turbulent environment caused by the aeration makes it difficult for the rotifer to reproduce. In biofloc, rotifer usually does not reproduce in large number. As long as you keep the operation optimal, you can already observe the rotifer under a microscope. Next is the nematode. Nematode presence in low quantity at the early stage of biofloc. The count becomes lesser due to grazing by the shrimp. Towards the end of the culture, when the shrimp size becomes bigger, they will graze on bigger microorganisms like the nematode. Consumption of nematode by the shrimp helps to improve their nutrition requirement. The nematode represents an important source of proteins, fat, and carbohydrate for the shrimp. Although some people say that the nematode is a pathogenic organism that causes infection in many fishes, research has proven the nutritional quality of this organism for the shrimp. Some other studies also reported that the highest post-larvae turnover rate was achieved when the larvae were fed with the live nematode. The other common microorganism in biofloc is algae. Algae is one of the most diverse aquatic organisms that are able to conduct photosynthesis. Seaweed also a type of algae which is usually consumed by humans. On the other hand, algae in biofloc are microscopic, where it only can be observed under a microscope. They also can present in various shapes and species. The algae in biofloc, they function to assimilate ammonia and potentially phosphate and nitrate. In the presence of sunlight or artificial light, they will undergo photosynthesis that will eventually accumulate oxygen level inside the biofloc system. As the biofloc system required a high dissolved oxygen level, the net production of oxygen by the algae also important. However, the amount or count of algae in the biofloc system should be at the optimum level. This is due to the drawback of algae that can bloom and crash. Also potentially develop harmful algae bloom (HAB). If the biofloc system change toward green color, the system potentially exposed to oxygen crash, especially during the night time where the algae only respire at night. So in this case, farm operator should understand the loading and removal of the solid in the biofloc system so that algae do not dominate the water. Lastly, copepod. Copepod is well known as a traditional life feed of the fish larvae and shrimp larvae. 13,000 species have been recognized, which include the marine, brackish, and freshwater species. Copepod is a popular choice among the farmers due to its nutritional superior. It is rich in protein, lipid, carbohydrate, and enzyme, which help in the survival and optimal growth of the animal. For the shrimp, Copepod is also the source of astaxanthin, which is a compound that responsible for shrimp pigmentation. Aside from all of these good microorganisms, there is also bad microorganism in Biofloc. This bad microorganism or the undesirable microorganism called as the filamentous algae. If you have this filamentous algae, you can see that the surface of your biofloc water will full of foaming. Something similar to this picture or even worse. Filamentous algae under the microscope look something like this. If there is a presence of fat and oil in the water, it will promote the growth of this filamentous algae. And it is unfavorable for the biofloc system as it will compete with the shrimp for oxygen and also reduce the water visibility. Some other bad microorganisms are virus, infectious bacteria and parasite. We usually send our sample to the lab for vibrio analysis using this TCBS plate, the green color picture over here. And the lab analysis was done as a precaution to avoid infection to the other tank or pond. Usually if we found out that our water sample from the tank is have high concentration of vibrio, we will have a quarantine procedure for that tank. To conclude this presentation, I would like to highlight that different microorganisms in the biofloc system have their role to maintain the water and the cultured animal itself. Although some farmers would not take the microorganism count as an important step in their operational tasks, I would recommend including this step so that you know what is happening inside the biofloc waters. For those who are interested, we conduct an online and physical course on indoor biofloc farming with the Vaname shrimp. These are the course syllabus including knowledge and understanding, business skill, practical session and networking. Our physical course will be held in Keluang, Johor, Malaysia for RM1,599 per pax. The course will be in 2 days. Usually we conduct on the weekend. Our next available slot is on the 8th to 9th of May, which is next month and in 2 weeks from now. However, for those who cannot join us in Malaysia, we also have an online course that is pre-recorded on our website for just $199. If the international border open and you want to visit us again to see the farm, you can have a full rebate on the cost, meaning you just pay the remaining fees minus the online fees. And then we have special forum for our online subscriber. We open a forum on our website for the topic so we can discuss further on the issue, or you can scroll down to the forum to see if anyone got the same issue with you and how do they solve it. So that's it for my sharing session. You can drop us your WhatsApp or email us any of your inquiry. Mr. Yit here will take over for the Q&A session.
So hi guys, is there any questions from the session? So I see some of the repeated participants. I see William, that's back. Sunil, I think not too sure What are you referring to, Mr. Sunil Kumar? Alright, so I'll just leave it there for anybody to ask any questions. So hopefully that's the presentation is clear on what to expect and what to monitor in terms of biofloc culture. Hi Alex, good evening. Alright, so I don't think there's any questions from the crowd. Perhaps if you have any questions, you can drop us an email at yet at this email. Can you drop us an email at sales@rs-aquaculture.com, and we'll be happy to respond to your email. So the next webinar will be held on the second week of May, which will be on the 12th. Right, the 12th of May. So do mark your calendars for our next webinar session. The topic will be is yet to be decided, but we'll announce shortly, probably a day or two after today. So if that's the case, so I'll see you in the next webinar session. So take care and stay safe. Wow, first time no question.